The next topic we're going to discuss is intracellular trafficking. Recall that before we spoke about membrane composition and movement of molecules through membranes, we discussed the processes of transcription and translation. Translation occurs in the cytoplasm, and there is in the cytoplasm a common pool of ribosomal subunits, small and large, that were synthesized in the nucleolus of the nucleus. The small ribosomal subunit is going to find an mRNA that needs to be translated, and when the start codon is encountered, a large ribosomal subunit will grab on, and the process of translation will continue until a stop codon is reached. Those proteins that are synthesized in the cytoplasm need to function in specific places in the cell. But how do those proteins get from the cytoplasm to the intracellular locations where they need to function? That same question was posed by Dr. Günter Blobel. Dr. Blobel was born prior to World War II in East Germany. Because of the war, his family escaped to West Germany, where he ultimately studied medicine. He left Germany in the 1960s and moved to the United States, specifically to Wisconsin, where he earned his PhD at the University of Wisconsin at Madison. His work there verified that indeed the signals for protein targeting or protein location were found within the polypeptide chain. In the simplest form, those targeting signals are stretches of amino acids known as signal sequences or signal peptides. These can be found in proteins that are in their unfolded state. Alternatively, there can be a signal patch found within a protein that has folded into its three-dimensional shape. You cannot, in these proteins, find stretches of amino acids that are continuous, but rather small bits that, when the protein is folded in its three-dimensional shape, form a patch. Inherent in the idea that there are these targeting signals in a polypeptide chain are some other considerations. First, there must be proteins that recognize these signal sequences or signal patches in the cytoplasm where protein synthesis occurs. In addition, there must also be receptors for these recognition proteins on the target organelle where the protein is meant to be targeted. Dr. Blobel verified that targeting signals are contained within the polypeptide chain through a series of rather ingenious experiments. Take, for example, the two proteins shown on the image on the left. There is a blue ampersand-shaped protein that is meant to be targeted to the endoplasmic reticulum, and contained within that blue protein is a signal sequence. There's also a green peace sign-shaped protein that is meant to reside in the cytoplasm. This protein has no signal sequence or signal peptide. What Dr. Blobel did was he modified the gene for the ER-targeted protein by removing or deleting the part of the gene that encoded the signal sequence. When he looked at where that genetically modified protein resided, he found that, in the diagram on the right, the ER protein with the signal sequence removed was found in the cytoplasm. This demonstrated that this sequence of amino acids was necessary for ER targeting. This was not the end of Dr. Blobel's experiments. He then took that bit of DNA that encoded the ER targeting sequence, and he transplanted it in the gene of the cytoplasmic protein. Therefore, the cytoplasmic protein, when synthesized, had this signal sequence attached. Where this protein ended up was, where this genetically modified protein ended up, was in the endoplasmic reticulum. This demonstrated that not only was this sequence necessary for ER targeting, but that this sequence is sufficient to get a protein in the ER. This table summarizes a bit of information that we'll be discussing. Looking at the targeting signal, the final destination of the protein, where protein synthesis completes, whether or not transport to the target location occurs when the protein is folded or unfolded, and the mechanism by which those proteins are transported. On the top line of this table, we see that in the absence of a signal sequence or signal patch, the final destination of a protein is the cytoplasm, where protein synthesis completes. Proteins who are completely synthesized in the cytoplasm can fold in their three-dimensional conformation, and when they do, they are subsequently able to move around freely about the cytoplasm by diffusion. If a protein has a nuclear localization signal, that protein is meant to be in the nucleus. These proteins are completely synthesized in the cytoplasm, and they are folded in the cytoplasm. Therefore, proteins that are meant to be targeted to the nucleus are transported into the nucleus in their folded conformation. They are transported into the nucleus via gated transport, meaning they must go through some sort of gate. In this case, the gate is the nuclear pore complex. I will, however, say that some proteins of sufficiently small size are able to move through the nuclear pore complex without the help of important proteins that transport those proteins with nuclear localization signals. I make the analogy here of the nuclear pore complex being like a wrought iron gate across a driveway. Certain things like rabbits and squirrels and butterflies and beetles are able to move across that gate without having the gate lifted. They are sufficiently small and compact. However, motorcycles, delivery trucks, cars, they necessitate the opening of the gate in order to move across that entranceway. This is the way the nuclear pore complex works. Small things can move through the nuclear pore complex without the gate opening, but larger things require the opening of the gate. These larger things must be transported through the gate via important proteins. In the next line of the table, we focus on mitochondrial localization signals. Mitochondrial localization signals are sequences or stretches of amino acids within proteins meant to be targeted to the mitochondria. These proteins are also completely synthesized in the cytoplasm, yet remain unfolded through the aid of HSC70 proteins that will carry these unfolded proteins to the mitochondria, where they will enter the mitochondria via transmembrane transport, specifically using Tim and Tom transport proteins, about which we'll learn a lot later. Proteins that are meant to reside in the ER or anywhere in the secretory pathway will have an ER localization signal and possibly some other signal within the polypeptide chain. As a reminder, this is an image showing you the secretory pathway. Proteins synthesized at the endoplasmic reticulum follow the pink arrows to the Golgi apparatus, move through the various stacks of the Golgi, and then enter transport vesicles, ultimately leading to exocytosis from the cell. An alternative pathway within this secretory pathway allows for proteins synthesized at the ER to travel through the Golgi and end up in an early endosome that ultimately will become a lysosome. This is the secretory pathway that any protein containing an ER targeting sequence will follow. Again, looking at our table, a protein meant to be at the ER or anywhere within the secretory pathway will have an ER localization signal, and that is a sequence. Proteins that have an ER signal sequence are not completely synthesized when they find their way to the ER. They will find their way into the ER lumen via transmembrane protein, and that transmembrane protein or the channel is known as Sec61. The transport protein that carries the partially synthesized protein to the ER is known as SRP, signal recognition particle, and we'll learn more about the specifics of SRP in future videos. After entering the ER lumen, of course, proteins travel elsewhere within the secretory pathway via vesicular transport. There must be some other signal within the polypeptide chain if a protein is meant to remain within the secretory pathway. In the absence of this additional signal, proteins follow the secretory pathway completely and end up outside the cell. These proteins also enter the ER lumen via transmembrane protein and find their way to the outside of the cell via exocytosis of secretory vesicles. This is a cartoon diagram summarizing what I just said. Proteins begin their synthesis in the cytoplasm. If those proteins contain a nuclear localization signal, they will be transported via gated transport through nuclear pores. If those proteins contain targeting signals for chloroplasts, mitochondria, or peroxisomes, those proteins will be completely synthesized in the cytoplasm and be transported into those organelles via transmembrane transport. Transmembrane transport is also required for transport into the endoplasmic reticulum, but proteins synthesized who complete their synthesis at the ER will ultimately enter transport vesicles and be transported to various locations within the secretory pathway or to outside of the cell. This is another way to depict this process. Rather than a cartoon diagram, this is sort of a flow chart looking at the same thing. We will begin our discussion of intracellular trafficking by focusing on transport into the nucleus, transport into the mitochondria, and transport into the ER. I've spoken about the mechanisms by which proteins are targeted to specific intracellular locations, but I haven't spoken yet about what those targeting signals are. This table shows you some examples of those targeting sequences. We'll not focus on targeting to peroxisomes or chloroplasts, but we will spend some time specifically looking at how proteins get into the nucleus, how proteins get exported from the nucleus, and how proteins get imported into the mitochondria. Import to the nucleus requires a nuclear localization signal, and nuclear localization signals are positively charged amino acids. Again, this can be a stretch of positively charged amino acids, or it can be a positively charged patch, but either way, those positively charged regions of a protein are going to be recognized by a protein known as important. Export from the nucleus requires an amphipathic alpha helix, where one side of that helix is hydrophobic. Specifically, the hydrophobic side of this alpha helix should contain some number of leucine amino acids. This amphipathic helix will be recognized by the protein exportin that will facilitate nuclear export. Import to the mitochondria also requires an amphipathic alpha helix, although this helix must be at the extreme amino terminal end of mitochondrial proteins. The portion of the amphipathic helix that's going to be recognized is going to be a positively charged side of the alpha helix. That positively charged side of the amino terminal alpha helix will be recognized in the cytoplasm by an HSC70 protein. Import into the ER will be our next focus of discussion, and this requires an alpha helix at the extreme amino terminal end of a protein, but this must be a completely hydrophobic alpha helix. The hydrophobic amino terminal end of a protein, when recognized by signal recognition protocol, will halt translation so that the partially synthesized protein can be carried to the ER where protein synthesis will be completed. If the protein is meant to be retained in the ER, it will have an ER retention signal sequence at its carboxyl terminal end. This ER retention signal is 4 amino acids long. The single letter code for these 4 amino acids are respectively K, D, E, and L. Therefore, the protein in the ER that will recognize and retain proteins with this signal sequence is known as the KDEL receptor. This has been an overview of intracellular targeting. The next couple of videos will speak about the specifics of both nuclear and mitochondrial transport.